for those of you who are watching from home. Okay, so one way to think about this, remember we talked about how the number of protons in the nucleus determines how close the electrons are to the nucleus, right? And so remember that we can have 2 electrons in this orbit, we can have 8 electrons in this orbit, and 8 electrons in this orbit. And also remember, as the nucleus draws the electrons in, is that stored energy or released energy? Stored, right? So when an electron moves from a higher, take a different color. So if it moves and it moves closer, right, that is stored energy. And another way to say stored energy is potential energy, right? So we can, if we hug the electrons closer to the atom, then we're going to store energy. If the electrons move away, then we're going to release energy, right? So it's kind of like a magnet when you take them together. Oh no, wait a minute, that's opposite, sorry. I said that oppositely, it's like a magnet. Okay, so when you pull them apart, that's stored, sorry. And when you put them together, that is released. So this is stored, sorry. And this is released. I had to go back to my analogy
so
of.
If electrons move closer to atom, it's released.
Yeah, so I have to go back to my idea of a magnet and opposite poles, right? She was right. So like if you move the magnets closer together, right, you're releasing the energy. And if you move them apart, it's like, right, there's stored energy. And if you let the magnets go, they can go, right, and back together. So that's how I always remember it. I always have to go like this, magnet. Okay, I'm storing energy if I pull it apart. I'm releasing energy if I put it back together, okay? So one way to view oxidation reduction is also in the hogging of electrons. So when we gain an electron, we could hog the electron. So they call it the hogging of electrons, right? Okay. So we might not gain it, it just might be hugged closer to and further away from the other molecule, okay? And so this is, would be not the hogging, but this would be kind of the release of the electrons. Okay, so I'll put this releasing, hogging of electrons so that they go closer to one atom than in the other. And we saw this in water, right? Because water is polar. So which atom here is hogging the electron? Oxygen, right? Because it's stronger, so it's hogging it. And the one that's releasing the electron is hydrogen, okay? So that's another way that we can think of oxidation reduction. Another way that we can think about it is in terms of losing or gaining hydrogen atoms. So instead of losing electrons, we can think about the loss of hydrogen, okay? And then this would be the gain of hydrogen. Okay, so reduction is gain, oxidation is loss. Whether it's just losing it a little bit, kind of goes further away from you, or whether you actually lose it and it's in the form of a hydrogen atom. And remember that the hydrogen atom just has one electron, right, attached to it. So it's just like a -1, right? It's got one electron. Okay. Okay, so let's look at an example of an oxidation reaction. And this is something that happens in your body, right? So we have that glucose molecule, and we can combine this with 6 oxygen molecules. Get this chemical reaction that goes in 2 directions. So this can produce 6 water plus 6 CO2 plus ATP. Okay. So the chemical reaction that's going in this direction, in this particular instance, it uses oxygen. It doesn't have to use oxygen. I'll give you another example in a minute. So in this particular instance, it's using oxygen. And so we say that glucose is oxidized because the carbon, glucose, the carbon in the glucose molecule is losing hydrogen ions. So notice how here it doesn't have any hydrogen ions associated with it. So we see that glucose is oxidized down into carbon dioxide. Okay. So glucose is oxidized. So I'll put oxidation going in this direction. And then I'll put reduction going in the opposite direction. So we could say that carbon dioxide is reduced, which means that it gains hydrogen ions, right? And so it becomes glucose, okay? So this chemical reaction, I don't think I've mentioned this direction before. Maybe I did in the online lecture, but the energy in this case is sunlight, right? So this would be photosynthesis, right? This right here is cellular respiration.
So the reduction would be the photosynthesis and the...
No, yeah, reduction. So going this way is photosynthesis. So the way that plants are able to capture carbon is that they breathe carbon dioxide in, and then they reduce it to sugar, okay? Because they add, the reduction is a gain in electrons, but in this case, it happens to be hydrogen ions, okay? It's a little bit more complicated than this, but that's where we're gonna just stop right there, okay?
Just so change just, yeah.
Okay. I have another example that I'm going to use. Okay. Okay. This is a molecule, an organic molecule that's called something like nicotianamine dioxide or something like that. You don't need to know, just know NAD. We're going to talk about the role of this. But NAD can combine with hydrogen. So it can actually be reduced and it can actually store energy. So let me get my notes out. So NAD can combine with hydrogen ions, right? And produce what is called NADH. So this is a chemical reaction that goes back and forth depending upon whether we're storing energy or whether we're releasing energy. So in this direction, would this be oxidation or reduction? In this direction?
Oxidation.
Well, it's gaining a hydrogen.
So it'd be reduction.
It's reduction. Excellent. Right? So it is reducing.
It sounds funny that you're saying gain, but it's reduced.
I know. Reduction is gain. Oxidation is loss. Okay? It's confusing. So this one would be oxidation on the other side. Okay? So what I want to let you know is that when it reduces, it's actually storing energy. So this would be like a positive G, right? It's exergonic, it's storing, it's endogonic, it's storing energy. So this stores energy. Just like ATP, it's another molecule that stores energy. This oxidation, right, goes in the other direction, and it would have a negative delta G, and it would be releasing energy. Okay. So storing and releasing energy are what molecules do. And this is actually a really important molecule to the mitochondria. It's one of the ways that they actually, it's produced during cellular respiration, and it's one of the ways that it actually leads to the production of lots of ATP, okay? So that's an important example of a redox or a reduction oxidation reaction, okay? Okay, so let's look at enzymes. So enzymes are generally proteins, right? And the type of proteins they are globular. So they have a complex 3D structure. Okay, complex 3D structure. It used to be thought that it worked like a lock, and then the substrate was the key that fit in. But now they actually think that when the substrate binds to an enzyme, it actually changes the whole configuration of it, right? And so I'll show you an image of that in a second. So it has substrate binding sites on it. One of the interesting things is that enzymes are not broken down or used up during a chemical reaction. They can bind, cause the chemical reaction to occur, and then they're broken away, and then they can bind again, right? So they're not used up in a chemical reaction. Right? So if we look at a diagram of an enzyme, oops, where's my picture? I think I was being clever and putting it on that. Oh, it won't go back. Just a second. Respond. There we go. There we go. Okay. Okay, so this is my enzyme. So if I were to like take the labels off here, you would know that this is the enzyme. So it does kind of look like a lock and the key right here, right? In this particular example. The substrate binds to that active site, that binding site. And then what it does is it changes, everything kind of changes slightly, and then it breaks it apart. And so at the end of the chemical reaction, you have the products being released, okay? So what enzymes are said to do is that they catalyze chemical reactions. So that means that one thing that they do is they speed them up. Okay? They speed up the chemical reactions. The other thing that they tend to do is that they reduce the activation energy needed for the chemical reaction to occur. Right? They reduce the activation energy. So in most cases, in this particular example, breaking something apart, right, takes a little bit of energy to do that. And so when we look at the activation energy, these are the reactants, right? These are the products. And this is the energy that it has to get over. So it takes more energy. So you could probably heat up the enzymes, which also increases the energy to get it closer to that point where it can actually produce the products. But notice that if we add an enzyme, it reduces the amount of energy that is needed, okay? So that's the reaction with the enzyme and reaction without the enzyme. So sucrose, if we just put it in water, might break down a little bit if we heat it up, right? But it breaks down much more quickly if there's an enzyme present, right? The rate of reaction takes place much more quickly, so it catalyzes it, okay? Okay, so we can regulate how and when enzymes work. Because if you think about it, if you're producing enzymes, you sometimes want things broken down or built, but other times you don't. If you're a cell, you want certain enzymes to be active and certain enzymes to be inactivated. And so we have what is called inhibition, right? So this prevents enzymes from working. You inhibit their ability to do their job, their work. And you can do that through what is called competitive inhibition. And this means that a molecule would bind to the exact same binding site, so it would compete for binding sites with the substrate, okay? So an inhibitor, which is another molecule of something, talk about some drugs that are like this. Inhibitor binds to the substrate binding site. So it's actually competing with the substrate for that particular site. Okay? An inhibitor binds to the substrate binding site. Okay. Yep. We also have non-competitive. And this means that the inhibitor binds elsewhere on the enzyme. And it inactivates it. Enzyme. Right, so that's non-competitive. Both of those shut down enzyme activity, so reduce the amount of enzyme that is actually catalyzing the chemical reaction. So when we look at this, we can see that this is inhibition.
And it inactivates.
Inactivates. Okay. Inactivates it. Okay. So there's a term here which is called allosteric, which simply means that it binds to a site other than where the substrate would bind, right? And so in this particular instance, the inhibitor will bind to a part of the enzyme and it changes the shape of the enzyme so it no longer can bind to the substrate, right? So that's called allosteric. So we're going to write allosteric next to that non-competitive. Okay, so this one is called allosteric inhibition. Okay, we also have the ability to increase the rate at which enzymes act, and so we have activators. Right? So we can have molecules that bind to the enzyme and actually activate them. So molecules bind to the enzyme to activate it. So if it's not active, they can bind to activate it. Now, the activators are really interesting because a lot of these are vitamins. So for example, when we talk about vitamins, sometimes they're said to be coenzymes. Right, so sometimes vitamins actually will bind directly to an enzyme to get it to work. Sometimes they bind to something else, which then ultimately causes a change in the enzyme, right? So a good example of this is vitamin C. So when we're producing a protein called collagen, which is found in our skin and our bones, vitamin C is the coenzyme to that enzyme that produces collagen, okay? So this is necessary for collagen production or collagen synthesis. And we talked about collagen because it is a type of structural protein, okay? So vitamin C is an activator. It is a coenzyme. We also have things like cofactors. And where coenzymes, like vitamin C, these are organic molecules, right? So they're composed of carbon, that carbon chain, right? So vitamin C is an organic molecule. Cofactors are inorganic molecules. So they would be like minerals. And they do the same thing. They can bind to the enzymes and cause them to become active. And so some inorganic molecules would include like iron, right, and magnesium. Those are both inorganic cofactors. So we need iron in some chemical reactions, right? I think this is a flip button. Anyway, and we also need magnesium. And there's other stuff we need as well, okay? So if we look at these activators, right? The activator binds to the enzyme. Notice how it doesn't have a, before it binds, iron and magnesium. It doesn't really have the shape, right, that it needs to have. But once we bind that, like once vitamin C binds to this, for example, the shape is changed, right? Okay. So that is an example of allosteric activation because it binds and then it changes the shape of the enzyme so that the enzyme can then bind to the substrate. Okay. Now another way that the production of or the action of enzymes can work is through, sorry, I have one more, through what is called a positive feedback mechanism. So this is separate from activation. Okay, so the positive feedback mechanism. So we talked about homeostasis a little bit. Homeostasis does not work by positive feedback. It actually works by a negative feedback mechanism. Okay. But this is an example where the substrate that is produced, or the product that is produced why is that positive feedback? Is that in your, does anybody have their book open? It says negative. Oh, sorry. Why did I put positive in my notes? Sorry, negative. I'm like, this is not right. Negative feedback mechanism. Okay. So the product that is produced inhibits the enzyme. Okay. So notice how here the substrate is broken down, and it's actually a series of steps. So it's like this process, we have 3 enzymes, right? And it eventually, when we get these, they're called enzymatic cascades. So one enzyme produces a substrate, then is broken down by another enzyme, which changes into a substrate, which is broken down by another enzyme, and finally we get this end product. And then notice that this end product would come back here and shut this all down so that this cascade of enzymatic events does not take place.
It actually just says feedback inhibition. It doesn't say negative on that picture. It just
Okay, well, inhibition means
it's just
negative, right? Yeah. Yeah, so thank you about that. So if it's inhibition, it would be negative rather than activation, okay? So the end product shuts it down. So in this particular instance, we have, and I can just draw this. So A gets converted to B, gets converted to C, gets converted to D, right? And this is the final product. And when we see these are sometimes referred to as enzymatic cascades. And one thing that's an enzymatic cascade in your body is your clotting of your blood is an enzymatic cascade. We have all these different clotting factors and enzymes. One clotting factor serves as an enzyme to produce another clotting factor, and then ultimately your blood will clot, right? So these are chemical reactions that will produce a final product. And generally, when we look at these, this one, when you look at the way that these enzymatic cascades evolve, this one tends to be the first one that occurs in the evolution of the chemical reaction. And then as we get further from the final product, like A being converted to B and then B being converted to C, that occurs after. But first we get what we need, so we need that final product, okay? So then in this final product then shuts down, right, you could do a feedback here, so it would shut down the production of A. Right, so I'll just put a little cross hatch through there.
So then, when you say A, which one on that would be technically A?
The green.
Okay, so that's gonna be, it's kind of like the whole process of where it hits it until it connects. to
Yeah,
it,
so this would
then
be
one-
the A, this is the green one, B would be the purple,
Okay.
C would be the blue, and then D would be your final product.
And then you can do the final product.
Yes, so that final product, right, can feed back and it can shut down the production of A, okay? So that's what's called a negative, it would be feedback inhibition. Okay, so another, in your textbook, they call this feedback inhibition.
So the end product basically just replaces the substrate and stops the process.
It doesn't replace it because it's not binding to the exact same spot. So it would be that example of allosteric inhibition, right? Allosteric inhibition right there. Okay, because it's not actually competing for the same binding sites, but notice how it changes the shape of that enzyme so that the substrate cannot bind anymore. Okay, so we see these enzymatic cascades, and we're going to talk about one of these enzymatic cascades now. Okay. Oops. Okay. So we're going to talk about metabolism of sugar. And so when you look at this word, glyco, we talked about this before, that means glucose, it means sugar. And lysis means to break apart. And so this is a type of breaking apart of the glucose molecule that starts off as not requiring oxygen. So if we look at glycolysis, it is anaerobic at first, right? So glycolysis is actually anaerobic, but the products of glycolysis can enter into aerobic respiration. I'll mention that in a second, okay? So this is glycolysis, it's anaerobic. It occurs in the cytoplasm of the cell. And sometimes you hear cytoplasm as referred to as the cytosol. So think of water with lots of things in solution inside the cell. So it occurs in the cytoplasm of the cell. Okay. It is found, and it occurs in both prokaryotic organisms, so that would be bacteria, and eukaryotic would be just about everything else, right? So that would be plants and animals and fungi. So it occurs in both of those. Now, in order for glycolysis to occur, it requires the input of ATP. So it requires 2 ATP molecules, but it produces, oh geez, I had this down. I'm now way ahead of my notes. Find my glycolysis. Okay, but it produces 6, and so there is actually 4. So it produces 4 ATP, sorry, 4 ATP. So that means that there is a net 2 ATP produced. Okay. So as I mentioned before, some chemical reactions require a little bit of energy input, and then they go, and then they release it. And so the delta G would be positive here, right? It's actually a positive value because it is producing a net of 2 ATP. Now this is not very efficient. And we're going to look at what happens when we take the products of glycolysis and then put them into what is called the citric acid cycle, and then we can produce a lot of ATP, but it's not very efficient compared to the other one we're gonna look at, okay?
When you say efficient as in how, like,
It's not producing a lot of energy.
oh, okay.
So for glucose, for breaking down glucose, this isn't a lot of energy, right? We can produce a lot more through another series of chemical reactions. Okay, so let's say we start with glucose, and we know that glucose has 6 carbon rings. 1, 2, 3, 4, 5, and I'm just drawing it out as a strand, because the first thing that the enzyme does is it breaks the ring and it makes the carbon come out into a strand of carbon, okay? So then we have 2 ATP added, and that puts energy in, and that gives us 2 ADP, right? So remember that ATP has this high energy phosphate. Adenosine triphosphate has 3 phosphates, right? Adenosine diphosphate has 2, right? So we have just lost 2 phosphate groups and we have released energy, right? So the way that this looks is that it looks like this. We now have phosphate attached to this molecule. 1, 2, 3, 4, 5, 6. So here's a phosphate group, and here's a phosphate group. Okay, so I'm going to put my phosphate groups in a little circle. Okay, so I'm going to circle my phosphate groups up. Okay, so that took energy to put those phosphate groups onto the carbon skeleton. Now what we're going to do is we're going to break this down into 2, 3 carbon molecules. So this would be phosphate, and we have carbon, 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 and we have phosphate, and we have carbon, carbon, carbon. Okay, so when we do this, this releases energy, and we're going to convert these into what is called pyruvate. So these are going to be converted to pyruvate. And this chemical reaction is going to yield us, this one is going to yield us, we talked about NAD, so I'm going to write my NAD here, positive, and this is going to gain, and it's going to be reduced to NADH. Oops, NADH. Okay. So NAD gets a hydrogen, and that means it's storing energy. And so this is a release of energy rather than energy being used, right? So this is a storage of energy, okay? And then we're also going to get ATP. And so we have, in this particular instance, 2 ATP, right? And I kind of ran out of room. Sorry, ADP. ADP, and we're going to do 2 ATP. Okay. So that whole arrow, I could draw that arrow longer, right? So this is the part of glycolysis that produces energy. The first step is the process that uses energy. So this produces energy. Right? And the thing about it is it also produces it over here. So you could draw the same exact thing over here, right? So this one produces a net, there's 2 ATP, 2 ATP. So that is actually 4 ATPs, right? But remember that I had to use 2 ATPs. So that is minus 2 ATPs, and that gives me, right, a positive 2 ATP. Okay. Now in your book, the diagrams of your blood. You can, if you're really, really curious, you can look at these diagrams and watch what is happening during this chemical reaction. Okay, so this is what it looks like in your book. Okay, so just notice, where is it using energy? You now know ATP is converted to ADP. It's using the energy. So it's using it to produce these molecules, and then it's going to eventually produce pyruvic acid. And the pyruvic acid is what we're interested in, right? The pyruvic acid or pyruvate. Now, we can talk about what happens in pyruvate in certain circumstances, and we can talk about fermentation. Now, when you ferment something, do you usually add oxygen or not? No, right? So fermentation is also anaerobic.
Don't you add, like, allow to have oxygen in one form?
When you ferment, it's generally, it doesn't require oxygen though. So the chemical reaction doesn't require oxygen. I'm trying to think about when I make beer.
That's right.
I guess you do have an airlock and it does breathe a little bit. Yeah.
No, you said, don't you allow it?
Yeah. Yeah, you allow it to breathe a little bit. Okay. But fermentation, the chemical reaction, does not require energy. Okay. So it is anaerobic. So this is where we take pyruvic acid or pyruvate, right? And I'm just going to put pyruvic acid, and we can use either one, right? And we're going to convert it to lactic acid. And we're going to do that in the presence of the NADH that we used. And so what happens is that NADH, which is a source of energy, is then reconverted to NAD positive. Okay, so this is lactic acid. So this is one type of fermentation, and this is actually occurs in your muscles. So think about when you do anaerobic exercise, like lifting weights, or sometimes if you do isometric, like maybe Pilates or yoga, right? You generally aren't breathing enough to use, to burn all of your glucose in an aerobic pathway. You don't have enough oxygen. And so this is what builds up in your muscles, right? And so this is a chemical reaction that takes place in the muscles. So this is lactic acid, okay? So this builds up in muscles. Right? It's also a waste product. Okay. And so it's just a way for, if we're talking about bacteria that ferment, it's a way for them to release, to break down pyruvate and produce lactic acid. So in terms of bacteria, lactobacilli, which are bacteria, right, convert lactose into lactic acid. So they actually release lactic acid. And you might have seen or heard of lactobacilli because this is what is commonly found in yogurt, right? And so this would be the fermentation of milk. So if you let milk ferment with bacteria, it produces a lactic acid. And then that pH, the drop in the pH, actually causes the proteins to denature in the milk, right? And then the yogurt becomes thick, right? So if you denature the proteins, they will come out of solution, and your yogurt becomes thick, okay? Also, when we look at some parts of the body, we have lactobacilli, and specifically, this is found in the female vagina. So these are good bacteria that lower the pH, right? So lactobacilli are found in the female's reproductive tract. They create an acidic environment, and the acidic environment helps to prevent yeast from growing. Right, so it prevents yeast growth. Right. It also kills weak sperm. Okay. So sometimes when females take antibiotics, it kills off these good bacteria that are normally found in the vagina, and then they get yeast infections, right? Because their pH of their vagina is not acidic enough to keep bad fungi and yeast out. Okay. So that is the chemical reaction that's taking place. Okay. So when we look at, this is from your book that shows the glucose, right? This is glycolysis. You get 2-pyruvate, and then you get 2-lactate coming out of that, okay? This is still anaerobic, okay? Okay, citric acid cycle. I'm not gonna finish, I'm gonna have to do a little recording after. But the citric acid cycle is aerobic. Right? So this means that it requires oxygen. Okay? It uses the products of glycolysis. And the products of glycolysis are the pyruvate or the pyruvic acid, you could call it, okay? Pyruvate. It doesn't occur anywhere in our cells except for in the mitochondria. So it only occurs in the mitochondria. Okay. It is much more efficient than anaerobic respiration. And in fact, it produces a net at the end, overall, it produces a net 36 ATP. Okay. And so that is actually not counting the 2 ATP that are produced during glycolysis. So if you take into consideration glycolysis, right? So if I put here, glycolysis produces 2 ATP. Then you could add 36 plus 2 and actually get to 38 ATP, okay? So sometimes people give this on a test. They say, okay, well, what is the net product number of ATP that are produced during cellular respiration? And they say 38, right? Or sometimes they say 36. The difference is just whether or not they take into consideration that first step, which can actually go either way. So fermentation is anaerobic, the citric acid cycle is aerobic, right? So sometimes it's 36, sometimes it's 38. So I don't know what to tell you on the test. Right? Hopefully they wouldn't give you the choice of those two, like if you were taking a test. I mean, not my test. I'm thinking of like when I took SATs or the ACTs or whatever, they asked what is the end product, right? And I think they want 38, right? I don't know. I actually sat down and tried to figure this out. Which one would be better? Okay, so when we look at the mitochondria have this weird internal membrane. So here's my mitochondria, and then it's got this internal membrane. And this internal membrane is called the cristae. And then inside this cristae area is the matrix, right? And the matrix is where the citric acid cycle takes place. Right? And the citric acid cycle will eventually end in just producing, as a byproduct, carbon dioxide and water. So it goes back to our cellular respiration where we break down glucose and we produce CO2 and water, okay? So the waste products of this one are not lactic acid, but the waste products are CO2 and water, okay? So we could get rid of extra water if we had to by urinating it out, if it's too much waste product, right? But generally we're actually underhydrated a lot. This we breathe out, right? So we can just simply breathe out the waste product of the citric acid cycle. Okay, so I'm gonna stop there and then I'll just do a little recording. You can look for it tomorrow evening, tomorrow afternoon, on what you need to know about the citric acid cycle.